So, good morning, everybody. We have got to one of the highlights of this meeting, the ISSLS Prize. My name is Helena Brisby, and I'm the chair of the committee for the ISSLS Prize. I'm going to start to give you a short introduction. Before that, we are going to look at a video that's been made about the prize. So just lean back and look.
In my early career, I've always read about the ISSLS Prize and the groundbreaking science in this paper, so I really felt inspired by the studies and their authors.
It comes from a society that's renowned for having exceptionally high standards, and it's one of the biggest and best prizes I think you can be awarded in spine research.
The ISSLS Prize program started in 2003, and it was a continuation at that time of a previous awards program in ISSLS called the Volvo Awards, which was initiated in the mid-1970s. And the ISSLS program is for lumbar spine research. It is in 3 different categories: basic science, bioengineering science, and clinical science.
The purpose of the ISSLS Prize, as was the Volvo Award, was to stimulate high-quality research on spinal disorders and problems. What is special about the ISSLS Prize relative to other Society Best Paper Awards is that it's given out for a full paper. Most other prizes are given to presentations during a meeting, but this is a full paper that is awarded.
Each such submitted manuscript is reviewed by anything from 6 to 10 or more expert reviewers, meaning that the submitted manuscripts undergo a very thorough and detailed review.
This is a truly international award, so it's not purely US, Europe, or Asia based. So it helps disseminating the paper around the globe, and it helps with that increasing the impact and the citations of the paper.
The monetary value of the prize is not to be underestimated. No one ever submits for that purpose, but it is one of the highest in the field.
And for many researchers, that means that you can continue to do research. There is not so many grant organizations giving money to spine research, so the money really means something.
Receiving the awards at a relatively young age, I felt gave me a certain credibility within the spine community, and it also opened up many doors to new opportunities to work with some of the leading researchers of that day.
It's still one of my highest cited papers, and it actually remains to be one of my highest cited papers even the last year. The prize has a lasting effect, and I think this is something I really appreciate about the ISSLS Prize.
For us, getting the ISSLS Prize has been highlights of our career, and it's a recognition for us from all the peer expert scientists, all the best of the people in the world for what we are doing.
So this video is on our web page, and please use it and inspire people to submit for the coming years for the ISSLS Prize. So now we move on to this year's prize. And since 2017, European Spine Journal has been the sponsor of this prize, and we are so grateful for this. We heard a bit about the history, and again, this is something that ISSLS established to recognize outstanding contributions to the field of lumbar spine research. And as you heard, it was initially named the Volvo Award and later became the ISSLS Prize. And for each year, 2 to 4 papers have been awarded annually. This means that about 150 papers have been awarded in the 3 categories: clinical, basic and bioengineering science over the years. And we also heard that many of these are highly cited papers, so important papers that are acknowledged. So for this year, we had 30 submitted manuscripts, and this is the same number as we had last year, a little bit more than 2021. And for each prize, the award is $20,000 US. And we have chosen this year to give out 3 awards, 1 in each of these categories. The papers are submitted to European Spine Journal and simultaneously to the ISSLS Prize. And I want to acknowledge Irene Sintel, the editorial office manager for ESGO, and Fiona Strain, our international coordinator, who has been doing a tremendous work to help us to get this together. We have had a prize committee, and you can read the names of this year's prize committee here. And I want to thank all the members on the prize committee, because it's quite a big job to do this in a good way, to review this manuscript really carefully. And when we have done that, we get the scores compiled and summarized by Fiona. But also, they are sent out to reviewers from the ESGO, and we get access to these reviews after our reviewing. And that means that each of the submitted manuscripts are read and evaluated by 6 to 10 people. Then we had a Zoom meeting in January and discussed these manuscripts and decided about the winners. And of course, we declare if we have any conflicts of interest and do not then participate in the decision about that manuscript. And these 3 winning papers are published now in May 2023 in the European Spine Journal. And also the other submitted manuscripts are considered for publication in the European Spine Journal. So I really want to acknowledge European Spine Journal and our member, Robert Gunsberg. I don't know where you are, but I think you are somewhere here. There he is, editor-in-chief, who's actually make this come through, that we can do this. So big, big thank you. So this year's awards then. I will tell you who the award winners are, and then we will take them one on one and hear their presentations and give out the awards. Slight change to previous years. The first prize in basic science, the name of the paper is Lactate in Lumbar Discs, Metabolic Waste or Energy Biofuel, Insights from In Vivo, MRS, and T2R Analysis following exercise and nimodipine in healthy volunteers. And Dr. Pushpa and Dr. Raisekaran will be the presenters, but you also have a number of good co-workers that have made this work become this good. So all of you, I think we give them an applause already now. And all of them that are here will also come to the stage later. The second one is in clinical science, and the name is Comparison of Degenerative MRI Features of the Intervertebral Disc between those with and without chronic low back pain, an exploratory study of 2 large female populations using automated annotation. And this will be presented by Dr. Fairbank, but also you have a lot of co-workers, and we thank you all for doing this good work. And the third award is given in bioengineering science, and the paper is named Age and Sex-Related Differences in Lumbar Intervertebral Disc Degeneration between Patients with Chronic Low Back Pain and Asymptomatic Controls. And the presentation here will be given by Aaron Fields, but you also have a number of co-workers, important ones. So applaud for all you. So it's not me that you want to see up here, but we are now moving to the exciting time to hear the papers. Each paper will have 10 minutes to present, and we will have about 8 minutes time to discuss each papers. And after this, for the first paper, I will ask all contributors that are present for that paper to come up and receive their awards. So I would like to ask, do we have that? No. Dr. Raisekaran and Dr. Pushpa to come up and give their presentation, and we get your slides up. the stage is yours.
Thank you, Helena, and thank you to the Society for giving us this great opportunity to present our paper. We have pleasure to present this on behalf of all our co-authors who have no disclosure COI. The study was funded by the Department of Biotechnology, Government of India, and Ganga Orthopedic Research and Education Foundation. Now we know that the disc has an environment which is mechanically stressed, hypoxic, nutrient depleted, and acidic, and disc degeneration is so inevitable. But the exact initiator and risk factors are still unknown. And that's probably the reason that while the whole spine community has focused on regeneration, only a few treatments have progressed to clinical trials, and none are consistent and clinically available. The current problem may be not in just the gaps in knowledge, but also one of the critical events is, what is the point of intervention? MRI identifies disc degeneration only after matrix changes have progressed. And so probably we have already crossed the point of no return, and that's the reason why these treatments are unsuccessful. We know that degeneration will progress through initiation, molecular and metabolic changes, cell death, disruption of matrix, and degeneration. And MRI is way below to identify disc regeneration, and the critical missing step may be to identify at the level of metabolic changes. But can we do it? Probably magnetic resonance spectroscopy and T2 relaxometry may provide the key. Just for people who are on the initiation, we have gone a long step in imaging, and if this is all were first initiated in the brain, and so if this is the organ of the brain, MRI looks at giving us a morphology depending upon the proton density. DTA came next, which looked at the movement of molecules and gave us the track fibers. But magnetic resonance spectroscopy is way ahead because it gives us a spectrum of the molecules over there. Here you can see that 14 different molecules are actually shown on the extent to which they are present in a tissue. T2 relaxometry, on the other hand, shows the water content, and that is an indirect reflection of the hydration of the disc. So the possibility of these novel radiological sequences to analyze the proteoglycans, collagen content and hydration, and also lactate and the allied molecules gave us the opportunity of having the possibility to estimate in vivo a real-time metabolic status assessment, ability to repeat it at multiple points of time, non-interventionally and safe. And we decided to do it in two diametrically opposite biological situations. One is a high energy demand, where the MRS and T2R was 10 in volunteers before and after a short burst of low back exercises, and this we know does cause some changes in the disc. And the second was a diametrically opposite situation of an increased diffusion and nutrition supply. In 2008, we had demonstrated that oral nimodipine, a calcium channel blocker, will give rise to high nutrition. And so we did this, and this study actually produced interesting results. I would like my co-author, Dr. Pushpa, present the radiological part of it.
Thank you, sir. So the study was performed in 3 phases. The first phase included the standardization of imaging protocol, where 10 asymptomatic volunteers aged between 20 and 50 years had routine MRI, MRS, and T2 relaxometry of all 5 lumbar discs. So in spectroscopy is represented in a graphical pattern. We could identify 14 different molecules in the lumbar disc. You can see the x-axis represents where the particular molecule will resonate. So it is quite constant for a given molecule. For example, proteoglycan resonates at 2 and lactate is seen at 1.4. So this is constant for any given molecule along the x-axis. It is represented in part per million in chemical shift language. The y-axis is quite variable. It represents the concentration of the molecule in the disc. So you can see the structural molecules like proteoglycan, non-acetyl aspartate, and carbohydrate are the peak spectrum which we are seeing in the normal disc, whereas lactate, alanine, acetate, and propionic acid are the metabolic molecules which we are seeing as a lower end in the concentration. T2 relaxometry, on the other hand, gave us an estimate of water content in the disc. So it can be post-processed to give a color-coded images like this one, or we can simply draw an ROI and get a quantitative estimate of the hydration of the structure in question. So you can see here in the normal disc, the nucleus pulposus is quite hydrated and it is color-coded in orange, whereas annulus is in blue and greenish hue. Degenerated disc, on the other hand, shows a nucleus pulposus showing decreased hydration. So the normative data of these volunteers is already published in our European Spine Journal publication. The second phase was to study the effect of short-term targeted exercise on lower back. Again, we included 20 volunteers for this study, and we obtained the pre-exercise values followed by short burst of intense targeted exercise to the lower back, which consisted of flexion and extension of the lower back under resistance and for 5 minutes, and climbing of 150 steps twice. So following which we obtained MRS immediately after exercise and 1 hour post-exercise. So both T2 relaxometry and MRS were performed on these people. As expected, we did not find any change in structural molecules, that is proteoglycans and NAA and carbohydrate did not show any change in MRS, and T2 relaxometry also did not show any change in hydration. What was more interesting for us was the change in lactate and rest of the metabolic molecules. So there was immediate dip, like more than 50% dip following immediate exercise in lactate and rest of the metabolic molecules. And following 1 hour of exercise, there was gradual increase to the normal levels. So here in this example, in the Firman grade 1 disc, you can see the pre-exercise lactate level is around 2.6. It actually drastically decreased immediate post-exercise to 50%. The lactate is 1.5%. And following 1 hour, you can see there is gradual trend of normalization where it increased to 2.3. So there was a trend of normalization post 1 hour of exercise in majority of Firman grade 1 discs. So this is what the trend we saw in Firman grade 1. As compared to this patient here, where he had the grade 2 disc, the pre-exercise lactate level was 5.1. It did show drastic reduction immediate post-exercise to lactate showing 3, but it did not show a normal trend. So we don't see normalization 1 hour post-exercise, which we were able to see in majority of Firman grade 1 discs. So what was another interesting fact we came through is with increase or decrease of lactate, there was corresponding changes in propionic acid, alanine, and acetate. This gave us an idea whether they are involved in a similar metabolic pathway. The third phase involved the effect of increased oxygen and glucose concentration on disc, for which we enrolled 10 volunteers who took 1 week of nimodipine. So we obtained scan before intake of nimodipine and after 1 week of intake of nimodipine. Again, we did not see any major change in disc hydration. As you can see, the T2 relaxometry of the disc was 138 before before the nimodipine, and following post 1 week of nimodipine, it was 144. So there was no major change in disc hydration, and also in the structural molecules in the MRS. So proteoglycan and NAA did not show much change in MRS. But what again was surprising was that the lactate and rest of the molecules like propionic acid, alanine, and acetate showed significant increase following 1 week of nimodipine. So this is again a Firman grade 1 of a normal volunteer where pre-nimodipine lactate was 1.5. It showed drastic increase following 1 week of nimodipine to 5. So this was the major finding what we saw in Firman grade 1, but it was absent in Firman 2 and 3 grade discs. Again, the effect was more common, more evident in less than 30 years of age group as compared to 40 years and above. So Of all these findings, one of the things which actually catched our attention was around 16% of the exercise group and 12% of drug group with Firman grade 1 disc showed abnormal lactate response. And we think these may be the subset of Firman grade 1 disc which are showing abnormal nutrition or abnormal metabolic pathway. And we are hoping that these could be our target for preventive therapy. Now I pass on the discussion to Dr. Rajshekharan. Thank you.
Thank you. So, yeah, so we are all looking at diagnostic biomarkers for early disc degeneration. And we know that clinical is so confusing and vague. Radiological identification is much delayed. And so the question that comes begging is, will metabolic biomarkers actually do that? There's a lot of discussion not just on the disc, but in every other tissue, functional tissue, and lactate has been discussed and is the queen of the molecules. However, the traditional view is that lactate is a metabolic waste. It is a dead-end waste product and considered to be a criminal because it decreases the disc pH, suppresses proteoglycan matrix synthesis, increases cellular senescence, upregulates interleukins, and increases MMPS activation, leading to disc degeneration. There is ample literature support for this. In fact, it has been suggested as an effective biomarker for degeneration. And this is a famous equation which we always read. Lactic acid plus nerve is equal to pain. But our study actually shows that lactate may not be just a foe, but it can also be a friend. And the concept of lactate shuttle has actually come into being. The main issue is that we always thought lactate to be participating only in glycolysis, which means that it is an end product, and it just gives 1 ATP of energy. But however, we know that lactate can actually be the beginning of another cycle, TCA cycle, oxidative phosphorylation, where it actually gives 16 ATPs over here. Now, this has been proved in solid tumors in the brain, where at the center of the tumor, lactate actually functions in the glycolysis. But actually in the periphery, this lactate is utilized in the oxidative phosphorylation for the actively growing oncogenic cells in their metabolism. Now, if you look at the annotation here, we can see that the center of the disc, where we think the degeneration is happening, is again a very hypoxic area. So the lactate metabolism may be there in the glycolytic pathway, but this could be actually utilized by the periphery of the annulus to have it in the TCA cycle. This has been suggested by Jill Urban and also Wang et al. In 2020, as actually showed in FEM studies that this can happen. So is there a further proof of this concept that is actually happening in the disc? So our findings are contradictory to the normal thought process, because in high energy demand, in places where you're doing a short burst of exercise, you normally expect the lactate to increase, but we actually found it to be decreasing, which means that the lactate is being utilized. And again, in the high energy supply and increased nutrition of nimodipine, you think the lactate will go down, but we actually found a surplus of lactate. But the most important evidence is that if lactate was only participating in glycolysis, then we will not have the other amino acids like alanine, glutamine, and glutamate, which are actually the parts of the TCA cycle. In our series, we found that whenever the lactate was going up, and then you also found that the alanine, glutamine, and glutamate also was going, which thirdly, as Dr. Pushpa said, these changes were more prominently seen in the healthy and younger discs, which means that it is a metabolic issue. So the implications of this study has got high relevance. To conclude, lactate is just not a waste end product. We think that is also a rich biofuel in the disc. Lactate cannot be taken just as a biomarker for degeneration. And this is completely diametrically opposite to the traditional concept. Lactate and metabolic concentration depends on age, disc health, degeneration, and is also dynamic, which means that so many of these studies on lactate before, which are actually looking at 1 point time of analysis, we have to just question whether these can be accurate or inaccurate. Lastly, lactate response, and not just a one-time analysis of lactate, can be a clinical radiological biomarker. Because we have Firman-1 discs over here, which shows the normal response. And then when the degeneration comes over here, we have the abnormal response. But what is critically important is that within Firman-1, we have a set of population which are showing metabolic abnormality. So this shows that this can be a part of this, and what we mean is that the metabolic response, rather than one-time evaluation, can be an effective biomarker for earliest degeneration. Thank you very much for your attention.
So the paper is open for discussion. Please come to the microphone and introduce yourself before asking questions. I can see Dr. Yoon, please.
Raja, thank you for an amazing research that you presented for us. I have one clarifying question that I want to ask. You said climbed 150 steps consecutively times 2. So they're climbing a bunch of stairs. Is that what that is? So it's a very aerobically intense exercise?
Yeah, because the 150, because that's also quoted in literature, and because our hospital has got 6 floors, which has 150 steps. So the volunteers who formed the staff of the hospital, I mean, they climbed that.
How many of your individuals are able to do that successfully, or
yeah, you know, these
time
are
limits or anything?
Yeah, exactly. So we were targeting young, healthy volunteers, and you know, nowadays aerobic exercises, marathon running, and fitness is the mantra for this group of people. I mean, they were able to do it.
So you're suggesting that when you do vigorous exercise like that, that somehow the cells in the Firman grade 1 discs are able to utilize the lactate. That's what you're suggesting, which then would have to say that those cells in that disc somehow can recognize that there's activity going on and then is revving up the metabolism. But those cells, of course, are not involved in the muscular part of the exercise or anything like that, but it's just somehow responding. How do you think it's responding? What are the signals that tell these cells to increase their lactate metabolism?
Yeah. So Tim, this is a good question, but you know, it is actually taking us to hypothesizing or speculating what it is. I don't think we have come to that level. That will be the next step. But the importance of this study is that it shows a completely different response to what you would think. These people were undergoing a flexion exercise, then they were doing this running, and then they were climbing up the steps, which is a high energy demand situation. And you would think lactate will increase, because just like the lactate increases in the muscles when they do it, but actually we saw an opposite effect. And Wang et al. In 2020, actually they have done a FEM study model, and they have clearly shown that this is what is possibly happening, similar to a situation which is happening in the solid tumors. So we are proposing that we should not blindly think that lactate is a end waste product. So it's a fuel for thought. So I think much more studies need to be done on this.
I think we have to move on. There are so many questions. So we take alternative.
Ajay.
Raja, congratulations again, wonderful study. And my question goes a little bit in the same direction. I want to simplify a little bit more. Since since decades, we are discussing how do we have to do exercise, how do we have to move. And I thought we finally agreed that the diffusion process, which are responsible for the nutrition, are so slow that we cannot give a good advice. But now you show this exercise has an effect on the metabolism. Do we have to reconsider everything what we discussed in the past years?
Not fully, because here we are discussing 2 aspects of it. One is metabolism, and another is what you are talking about is hydration. And you know, metabolism was what was discussed in the phase 2 of our study, because lactate is using it. But nimodipine actually increased the nutrition. It increases the diffusion, and in the Eurospine Award paper, we showed that nimodipine increases the diffusion by 13% to the center of the nucleus. In the same situation, we find that the lactate is increasing, which means, I don't know, this is again speculation, because, so we hypothesize that in the situation where there is a lot of, with the diffusion, glucose and oxygen transport also occurs, so lactate is unutilized. And that is the reason why after nimodipine, there is an uptrend of lactate. Whereas in the opposite situation where they are undergoing a short burst of exercise, lactate is utilized, and that's the reason why lactate is coming down. It's a very fertile area for further study.
So we are not having so much more time, but I will allow 2 questions from each microphone. Try to be short and try to answer a bit short, please.
Gwen Soa, Pittsburgh, USA. Congratulations on this beautiful work. I had a technical question. When we look at these spectra, typically ex vivo, we'll commonly spike the sample with a known concentration of something to allow us to normalize across different scans. What are you able to use within an individual to normalize the concentrations that you're recording for lactate, for example, across different time points?
We used to do something called shimming during each cycle. So every patient used to have an MRI scan only after what is called shimming. So this will actually normalize the magnetic field as well as the protocol. So, and the signals from water is completely suppressed, and then we take the signals from the rest of the molecules. So it is purely technical, but it serves the purpose.
Thank you.
Hi. Excellent study. Congratulations. My question is, again...
Please introduce yourself.
Asim Sharma, University of Michigan. Technically, I assume this was a single voxel technique you used?
It was a single voxel.
I would have been very happy if you showed the reliability data on same individual for quantification using a technique that is so much influenced by the susceptibility effects. So do you have that data? Did you study?
We have a normal data on 50 volunteers. 50 volunteers, which we have not included in this particular study, but we have done a study on 50 volunteers on different time span, and we have found that there is no much variability based on the day or based on the same individual at different time.
So no difference at all? Like what's the normal range?
So once we do a proper water suppression and proper pre-shimming before the acquisition of the MRS, there is no much change.
I guess my question is more the placement of that voxel, because that is difficult. It's not automated. You're manually placing it.
Yes.
So as a radiologist, if I'm asked to place a voxel on a disc today versus tomorrow, I'm bound to be different.
So we tend to focus only into the nucleus pulposus, exclude the end plate, annulus, and the bone. So that is the specific instruction given to the technicians. And also, if the disc is less than 4 mm, we will not able to do the MRS.
Thank you very much. Yeah. Congratulations again.
So thank you, everybody. Great paper. Big applause. And I would like to ask all the co-authors of this paper to come on stage, please. Where do we have some photographers? Should we be there or here? We can be here because it's in front of us. Then. It's easier for them to be. We can be here. So congratulations. Yeah, but we have to give them all. Thank you.
You can have all those.
Thank you so much. Okay, we move on to the second paper then. Can we have the first slide? And I would like to call on Dr. Fairbanks to come up to present his paper in clinical science. Are you two presenters for this one? Okay, that's good. Perfect.
Amar Jamaluddin. Anyhow, thank you very much. Well, good morning, everybody. President and Chair, ladies and gentlemen, it's been a great honor and actually a wonderful surprise to receive this ISSLS Prize for Clinical Research for 2023 in this marvelous setting of Spine Week. These are our acknowledgements, and we've no conflicts to declare. Now, the background of this study is that most, if not all of us here, are aware of the long-standing controversies in the relationship between spinal imaging and spinal pain. This slide references some of the recent studies that have compared MR imaging in people with or without back pain. Those in the left column who found no convincing association, and those on the right who did. In this collection of studies, there's only one systematic review by Brinjiki and others in 2015, which did conclude that there was a small difference in favor of an association of symptoms and imaging. So our aims were to see if the relationship between degenerative changes and MRI and low back pain can be clarified by using automated imaging. And secondly, to do an exploratory study analyzing the relationship in age and spinal level in two large symptomatic groups, in red through this presentation, and one large asymptomatic group in green. The symptomatic groups were from Genodisc of 874 females recruited across Europe, and a contemporary group of 763 females recruited in clinics in Oxford. The symptom duration was more than 3 months, and mean ODI score in these samples was 45. The asymptomatic population came from Twins UK and were 701 females. We used the validated automatic imaging software SpineNet to report on Fuhrmann disc degeneration gradings and other degenerative features in all three groups. We compared prevalence of degenerative features between symptomatic and asymptomatic groups in relation to age for both the upper and lower lumbar spine. SpineNet annotation is based on routine lumbar spine MR sagittal T2 protocols using all the available slices. Disc degeneration was graded on the Fuhrmann system. Selective degenerative features, that was disc herniation, central canal stenosis, marrow signs, and end plate changes, were graded as present or absent. And SpineNet's been currently validated in 5 publications, which are listed at the bottom of the slide. So in this slide, there are 4 graphs showing average Fuhrmann grade versus age. So that's average of the 1 to 5 Fuhrmann scale. The bands represent 1 standard deviation. The top left graph, A, compares the severity of disc degeneration by age in the caudal or low lumbar spine, that's L45 and L5S1, in the females in the 2 symptomatic populations, which show little difference. The top right graph, B, compares the severity of disc degeneration by age in the rostral or upper lumbar spine, let's say L12 or L23, in the same 2 symptomatic populations, which once again shows little difference. The lower left graph, C, compares the severity of disc degeneration in the caudal lumbar spine, that's to say at L45 and L5S1, in the symptomatic population in red and an asymptomatic population in green, all females. Here, there is an obvious difference, most marked in the younger age groups, where the symptomatic population shows much more severe disc degeneration. And finally, in the bottom right-hand graph, D, the rostral lumbar spine compared with the same symptomatic and asymptomatic groups, there's little discernible difference. We selected an all-female populations for these studies because this is what was available to us in our asymptomatic comparator, which are few and far between. In these comparisons, we are trying to discover features that distinguish symptomatic from asymptomatic. This excludes the L3-4 disc and discs with Furman 3 grades, which do not do this discrimination. In these two graphs, we compared the odds ratio, plus or minus 95 confidence intervals, of minimal Furman, that's Furman 1 and 2, and severe disc degeneration, Furman 4 and 5, in the caudal lumbar spine between symptomatic and asymptomatic groups, pooled and by age in decades. There are marked differences in the prevalence of disc degeneration, both mild and severe, between symptomatic and asymptomatic, especially in the younger age groups. The confidence intervals only cross 1 in the oldest decade, and so the differences are not significant in that age group. So the presence of both normal discs and very degenerate discs can distinguish symptomatic from asymptomatic subject. This summary slide shows the prevalence data for disc herniation, central stenosis, that's B, and C, vertebral body bone marrow changes, which are related to modic changes, and D, end plate defects. In all cases, this is a binary annotation, either present or absent, because of the numbers in these samples. The distribution of disc herniation with age is maximal in the 4th and 5th decade of the caudal spine. The prevalence of degenerative spinal stenosis increases with age, mostly in the caudal spine. Caudal bone marrow changes are common in symptomatics. These marrow changes are strongly linked to severe disc degeneration. End plate change prevalence is in all cases low and doesn't discriminate so strongly in either caudal or rostral lumbar spine. So in conclusion, we've shown in this and previous reports that SpineNet has been validated in various different study designs. Using automated annotation, we can rapidly process large data sets and compare one with another. It's important to consider both age and spinal level when making these comparisons in degenerative features. Severe disc degeneration seems to be the key biomarker and strongly linked to other degenerative features. We found similar patterns in 2 different symptomatic groups. And finally, we should point out that 30% of the symptomatic patients have normal MRI scans. We have a number of limitations. This is an all-female, all-twin comparator group, and unfortunately, it is really difficult to find such comparator groups. There are small numbers in the younger age groups than we'd prefer. It represents many different MR machines and protocols, but that was what SpineNet was trained to deal with, and it definitely needs repeat studies to confirm or conflict with this data. So, can we find better comparator groups? This shows the reported prevalence of degenerative features of the internationally available cohorts without back pain who've had MR scans, and these are all with human annotation. We might expect that these groups to have similar prevalence of disc degeneration by age, but as is obvious, this is not the case. The reasons for this may be data. Unfortunately, publications do not always have easily available comparable data that include age, and spinal level. Our study has shown that pooling degenerative features from every level of the lumbar spine, which has been normal epidemiological practice for years, undoubtedly confuses the picture. But annotation, we believe, is a most important variable. And we think that human annotation of disc degeneration, in spite of them all saying they use the Furman grading system, which is based on the based on the interpretation of images printed in a single publication in 2003. So we propose that using SpineNet as a form of Esperanto is one way of exploring this problem, because it's clear that these annotators are in a different place. And finally, of course, there are biological and environmental factors. These data are collected from different populations, and other genetic and environmental factors may have influenced the variance of prevalence of disc degeneration. But of course, you can't find that out unless you've got the annotation consistent. Thank you for listening. Sorry, I'll leave that one.
So the paper is open for discussion. Oh, you're a fast runner, please.
Yes, I am. Tim Yoon from Atlanta, Georgia. Jeremy, over here, by the way. Yeah. Wonderful paper, thank you. I had a question. I saw that there was a difference in disc herniations and stenosis in your groups. How much of a confounding factor were those things in resulting in back pain? Because certainly people with those conditions would tend to have more complaints of back pain.
Yeah, so I think in some ways I've got to refer you to the paper where we have tried to put in as much information with supplementary material. But we did, what we did do was to try with a Venn diagram to try and show the overlap between these features. And there's no question that all of these severe symptoms are closely linked to severe disc degeneration. So it's not so easy, at least in these data, to separate the effects of those two. And of course, not all disc herniations are symptomatic, but I mean, you could see from those curves, there is quite a substantial difference.
But you did control for that, right?
Yes. Yes. Well, the control are the asymptomatic population, so that's what we're trying to compare.
Okay, please.
Thank you. Marin Korade, Coop University Hospital, Finland. Thank you for this wonderful presentation. I think you're going in a fantastic direction. Just have a suggestion or a comment, if I can. On our work on the twin UK cohort in 17, 18, and 19, we actually shown very strong association between end plate defect cross-sectionally with low back pain and also disc degeneration, and also a strong predictive value of end plate defects, you know, when we go longitudinally for low back pain, disc degeneration, other features, when we started grading the end plate defect like quantitatively. And we used the grading proposed by Professor Azashekaran. And so coming here back to the actual study, I think a limitation can be the binary annotation, because we're actually making, you know, the MRIs easy to be read by the system instead of having the system reading something that we cannot.
Yeah, I think I will pass the answer. Sorry.
Yeah. Do you plan to move back or to improve to quantitative annotation? Because actually, if we continue this way and we continue with binary, we may come to some clinical conclusion that will be in conflict with previous work, and we are actually doing a step back then.
So I think very much the objective is to try and get bigger data sets, and we don't have those. And so these are the mechanisms to try and gather them. I'll ask Amir to talk about this, because it is an important issue.
Yep. Yeah, so you're right. It's the main limitation for the end plate changes in our system right now is binary, but it's because the way we built the system was from Genedis. The labels that we have from Genedis were from Ian McCall, who's a great radiologist. So he graded them. He graded a lot of things. So because we weren't sure which one is the answer to back pain. So Furman, everything, stenosis. And for M-plate defects, he just lumped a lot of different things. Schmoll's note is in his definition of M-plate defects. So it's just presence, absence, and that's how we built the thing. And we just, because we know we wanted to do classification at the end, we did a detection system, which is probably not good enough to do your grading scheme that you suggested. So, but we are planning to add more. We are planning to segment the body, and then we can look at the M-plate defect in a bit more detail rather than just absent and present. So it's in the plan, it's in the works, but.
But do we need more data sets then to do this? I mean, we need a few more thousands annotated scans, or can we do it?
I think we move on to the next question. You can continue in the break.
Thank you.
Please.
Hi, Jeremy, Nadine Foster. Thank you. Can I bring the questions more to the clinical arena then? So I'm hearing more data are needed to train the system and to test this, but in the meantime, would you say that these findings are questioning where we're at in terms of clinical guidelines and the message to patients that MRI findings are not related to symptoms? How far would you go in attenuating that for clinical practice?
No, well, thank you, Nadine. That's a really important question. So at the moment, in everything that we're talking about here, it doesn't, at the moment, turn into a specific treatment. But we're all, in all our different fields, are looking for more specific treatments, and therefore we do need to stratify our patients. And I think my message to, as much as anything, to the community is that we should not throw away MRI scans saying it's of no value at all, because it clearly can discriminate two different populations. And also having a normal scan may also be a biomarker. But I think we are some way off converting that to saying this is a pointer to use treatment A or treatment B. But I think that's the direction of travel. I think it is sad. I mean, I think there are problems in how the epidemiology has been approached in trying to answer this question. And people have pulled all the factors together, and I think for the reasons that we've shown, that just conceals the message. So I think that is my message to the guideline people, is that they should not say MRI is useless, but maybe to do proper studies in clinical sets to see how good these markers are.
So one short question, and then we end this paper.
Thank you very much for the interesting work. My name is Muath from the University of Queensland, Australia. I'm just wondering, you said 30% of those who had symptoms had no degenerative changes, is that correct?
Correct.
So what do you think may be causing the symptoms? I'm just curious.
Well, I suppose in a way that's why this society's been here for such a long time, to try and answer that question. But I think what it has done is perhaps to allow the use of a normal MRI scan as also being a biomarker to say, well, maybe we need to, if we're going to pursue a biological explanation for it, to include other things. I mean, we've just heard from Roger that there are things going on in a normal disc with exercise, which could well be really important markers. So I think it's important to get what value we can out of the imaging now, but also to think ahead as to future applications that may arise.
Thank you very much.
I think we have to close the discussion for this paper. Thank you very much.
Thank you very much. Yeah. Thank you. No, Amir is the top of the list, so maybe right down the bottom.
Yeah, that's me. Yeah. Thanks so much.
So I'll put on those.
Thank you.
Where would you like? Yeah.
So we still have one exciting paper to go, and I would like to call Dr. Fields up to the stage to present his paper, bioengineering paper, please. Can we have his first slide up? Yes, please. So.
Good morning. Thank you, Dr. Brisby, for the introduction. On behalf of my co-authors, I want to thank the ISSLS Prize Committee for honoring our work. We have a terrific team at UCSF, and I'm grateful for everyone's contributions, but I especially want to acknowledge Noah Bonheim. Noah's a talented postdoc in my lab who led the study, and unfortunately, he wasn't able to join us here today. We have no competing interests to disclose. So the role of disc degeneration in chronic low back pain is controversial. Although there's a strong implication that degeneration causes pain in a subset of patients, the reality is that disc degeneration observed on MRI is also highly prevalent in asymptomatic subjects. And so the clinical significance of degeneration in any one patient is ambiguous. And in part, this reflects a challenge of distinguishing between what is physiologic degeneration and reflective of normal aging versus what is pathologic degeneration and may be abnormal for a given age. In both research and in clinical practice, the Fearman classification system is widely used to classify structural deterioration on lumbar spine T2 weighted images. However, prior to structural deterioration, discs undergo biochemical deterioration, including the cleavage and loss of aggrecan, elastin, and collagen, and the accumulation of crosslinks. And these biochemical changes are largely inconspicuous on T2 weighted images. And so as a result, current understanding of disc degeneration is really biased towards the terminal or structural stages of the condition, and much less is known about early stage phenotypes that are distinct from aging. So the overall goal of this study was to identify imaging biomarkers that distinguish pathologic disc changes that associate with pain from physiologic disc changes that typify normal aging. And so to do this, we used quantitative T1-row MRI in order to compare disc biochemical composition between patients with and without low back pain. So T1-row probes the interactions between water molecules and their surrounding macromolecular environment, and prior work from Ari Bordekar and Don Elliott demonstrated that lower T1-row relaxation times significantly associate with lower levels or lower amounts of glycosaminoglycan content in the tissue. And so T1-row values provide a continuous, objective, and non-invasive estimate of disc biochemical composition, like a virtual biopsy. So in this study, we prospectively recruited 84 patients with chronic low back pain from the non-operative spine clinic at UCSF and 54 asymptomatic controls. All of the subjects were imaged on the same 3 Tesla MRI unit, and the protocol included clinical fast spin echo images with T1 and T2 weighting, as well as a T1-row mapping sequence, which we used to calculate the mean T1-row relaxation time in the nucleus pulposus of the discs using an automated neural network for segmentation. We then used multivariate and mixed effects regression models to test for associations between T1-row, which was the outcome, and subject age, sex, spinal level, a study group, and the interactions between those factors. So here's a summary of the group demographics for the 133 subjects who completed the MRI scan. You can see that both groups had similar mean age and gender distribution, and as expected, the back pain patients reported greater pain and disability compared to their age and sex-matched controls. Mixed effects regression models accounting for multiple measurements per subject, as well as accounting for subject age and sex, showed that nucleus pulposus T1-row values were significantly lower in the chronic low back pain group, here in blue, indicating greater biochemical deterioration than in the control group, shown in gray. When pooling the male subjects from both groups and the female subjects from both groups, we found that the T1-row values were also significantly lower by about 11% in the females than in the males, and I'll talk about those sex differences here in a moment. T1 row values also depended on age and spinal level. So in general, T1 row values decreased by about 1 millisecond per year, and they were lower in the lower lumbar spine than in the upper lumbar spine, so greater amounts of biochemical deterioration in the lower spine. Now, there were significant 2- and 3-way interactions between age and level and study group, indicating that the relationship between T1 row and age actually depends on both spinal level and study group. So let me show you some of those results now. So these plots here show the relationship between T1 row values and age for each spinal level. At L12 through L45, you can see that T1 row values decline with age, and the relationships are statistically similar between the two groups of subjects. However, at L5S1, the age dependence of disc degeneration differed between the groups. So specifically, the back pain patients had significantly lower T1 row values, and the group differences were much larger in the younger subjects than in the older subjects. It's also interesting that in the control group here, T1 row values were strongly correlated with age at all of the levels, whereas in the back pain group, the age dependence of disc degeneration was also strong in all of the levels except for L5S1, where the amount of variance explained by age was relatively weaker. So this suggests that aging effects on the biochemical composition of the L5S1 disc involve a relatively uniform set of factors from which patients with chronic low back pain tend to deviate. Finally, we found high levels of biochemical heterogeneity in discs that were otherwise structurally intact. So the histograms here on the left-hand side of the slide show the distributions of T1 row values for all of the voxels in all of the discs belonging to a given Fuhrman grade. And so you can see that Fuhrman 1 discs had a much wider range of T1 row values compared to the discs in the later stages of structural deterioration or structural degeneration. So the takeaway here is that not all Fuhrman 1 discs are created equal, and in fact, one Fuhrman 1 grade disc may have measurable levels of biochemical degeneration or biochemical deterioration as depicted by T1 row MRI, and those differences in deterioration could be useful in phenotyping patients and in identifying discs with early degenerative changes. So to recap, we used the mean T1 row value in the nucleus pulposus as a quantitative biomarker of biochemical deterioration in the disc, and we found group differences both in the overall extent of degeneration and in the role of aging. So I want to stress here that these results do not provide evidence that pain in the back pain group was discogenic, but rather that biochemical degeneration in the back pain group was greater than biochemical degeneration in the control group, and that the difference between groups depended on both age and spinal level. At L5S1, back pain patients had substantially lower T1 row values than controls in age-adjusted models, suggesting accelerated biochemical deterioration in those discs. This also suggests that age-adjusted T1 row values could be useful in distinguishing degenerative changes that are pathologic and associated with pain from degenerative changes that are physiologic and associated with normal aging, particularly in the younger individuals. So to illustrate this point, I want you to consider a T1 row value of 60 milliseconds in a 30-year-old subject. Based on our study sample, that mean T1 row value, 60 milliseconds, would be 30% below average for a typical asymptomatic subject, but would be well within the range of T1 row values expected for 30-year-olds with back pain. On the other hand, by age 50, a T1 row value of 60 milliseconds would be completely unremarkable. In other words, it would be equally likely in patients with pain and in asymptomatic controls. So the extent of degeneration measured using T1 row biomarkers may be more relevant to clinical phenotyping when assessed relative to a person's age. Interestingly, we found that there were lower T1 row values in women than in men, implying lower amounts of proteoglycan. The mean age-adjusted difference was around 8 milliseconds, which is equivalent to the degenerative biochemical changes that would be expected to occur over a roughly 6 to 9 year period. So it's a substantial sex difference there. These findings are surprising because sex differences in lumbar disc health in humans are not commonly reported. At this point, the reasons for these differences are unknown. We do know that males and females show differences in lumbar spine anatomy, including differences in lordosis, differences in vertebral morphology, differences in muscle mass. It's possible that these differences either include or influence disc biochemical composition. And finally, there was a high amount of biochemical heterogeneity in structurally intact discs, Furman 1 and Furman 2 discs. So this heterogeneity could either reflect differing rates of biochemical deterioration occurring prior to structural deterioration, or differing peak levels of disc health. For example, Avico and colleagues recently showed that lower levels of disc health following the pubertal growth spurt were associated with back pain later on in life. So taken together with our current findings suggesting that the differences between back pain patients and controls was greater in younger individuals, I think the results collectively suggest that individuals may achieve different peak levels of disc health, for example, depending on either genetic or environmental or lifestyle factors. So in conclusion, physiologic disc degeneration at L5-S1 followed a relatively uniform course from which those with chronic low back pain tended to deviate. And as such, age-adjusted T1-Rho values at L5-S1 may be highly relevant to clinical phenotyping and to distinguishing pathologic changes that associate with pain from physiologic changes that associate with aging. Again, I want to acknowledge the study team that helped, and also our funding sources, and especially the NIH HEAL initiative for supporting the work. And I'm happy to take any questions. Thank you.
So, yeah, please, Dr. Eskan, please.
Yeah, Rajashekaran from Aaron, congratulations for this excellent study. I think both our studies reflect on the broad aspect. All Furman grade 1s are not the same. So thank you for resonating with our study. But to the question, so you said there are many biochemical differences which give rise to that. What do you think is the exact biochemical change that is happening? What are the changes actually that you think is causing this T1-Rho change?
Yeah, so in prior work that's looked at tissue samples and T1-Rho in the same subjects, primarily autopsy samples, the relationship between T1-Rho and glycosaminoglycan content is fairly strong. In this study here, I didn't show the results, but we have MR spectroscopy data, just like your group collected as well, and we showed that lower mean T1-Rho relaxation times were significantly associated with lower PG peak levels from the MR spectroscopy signature. So at this stage, I think we can confidently say that the amount of proteoglycan is significantly associated with the amount, or with the T1-Rho relaxation time, and so lower T1-Rho relaxation times reflect less proteoglycan. But I want to stress that there are other changes, for example, to the proteoglycans that could also affect T1-Rho values. For example, the extent of their biochemical cleavage. That's just one example of a biochemical change that might also affect the T1-Rho values.
Yeah, since you have both these values, MRS and T1-Rho, we also did an initial study taking both and chose MRS because it seemed to reflect better. So is there any reason why you chose T1-Rho over MRS?
Yeah, great question. So like you, we use a voxel-based MRS prescription, and it's a lot easier for us to delineate the nucleus pulposus using automated techniques from the neural network on T1-Rho, which is a spatial sequence, rather than using the voxel-based spectroscopy results. The other issue was we didn't have MR spectroscopy data for all of the subjects in the study, just about 50 of them. So
thank you. Thank you. Congratulations again.
Thanks a lot.
Please.
Great study. I noticed that the biggest finding that you found, I think, is that the L5-S1 differences in T1-Rho, and you know, the initial knee-jerk reflex is to think that, well, maybe this association means that you know, the loss of proteoglycan is correlated with back pain, i.e. causing back pain, but it could be interpreted a different way, that these people with chronic low back pain might have some other reasons, such as less exercise and so forth, and this is a effect, not the cause of things. So one way you might try to deal with this is to look at possible confounding variables such as activity levels and other things. And I'm just wondering if you think you might be able to do that in the future.
Yeah, great suggestion. We haven't looked at that yet. We have some biopsychosocial data on these same subjects, including a range of psychosocial questionnaires, so we can start to get at that. Probably not with the fidelity that you're suggesting, but I think it's a great point.
Yeah, Achim Wilke from Ulm in Germany. Aaron, congratulations, great study, very nicely standardized. Really, I respect that a lot. I would like to come back to the control group. Maybe you told that, maybe I missed that, but could you quantify that a little bit more or define that a little bit more? Were that patients who never experienced back pain or just not at the time point when they took the x-rays or the MRIs, or they are not chronic low back pain patients, then they correlated or they matched very well with the other group. So how did you do that?
Yeah, so the first part of your question is what is the nature of these controls? So these are patients that had no episodes of pain either at the time of the scan or historically. So they are not patients with repeated episodes of low back pain. So they don't have low back pain and they haven't had low back pain. So that's, I think, the first part of your...
But how did you find them? How did you...
Yeah, so...
What was the reason for the MRI?
Yeah, so we recruit through two methodologies. One is through flyers posted around the university and through local media. That's one way to do it. There's obviously challenges with ascertaining that these people did in fact not have back pain. The other way we do it is through an EMR query of our institution's electronic medical records. So we can basically set up searches through the electronic medical records to exclude patients that have either discussed with their physicians back pain in their clinical notes and back pain codings through ICD-9 and ICD-10 codes. So it's not a great way of doing it, and I think you bring up a really great point, which is, and I think Jeremy also alluded to this, very difficult to get control subjects for these studies. So,
okay, thanks so much.
So we are running out of time, so I will give the last question to Dino, please.
Okay, well, thank you. I feel so honored. Aaron, fantastic study, and congratulations to you and your fantastic group at UCSF. You know, this is fantastic because I feel like it kind of also validates some of the work that's been done with traditional MRI and looking at also different age-adjusted scores of degeneration that have been predominantly published in other larger population cohorts, epidemiological cohorts, mainly coming from Asia. I think two concerns that I kind of have is that with the population or the sample size that you have here, one probably needs to be very careful, especially when doing modeling, utilizing a string of different covariates to avoid overfitting the model. This may actually complicate things and can present certain noise. But the other I guess concern I have, not concern, just an inquiry, is that I've noticed, for example, in this study and even in the first presentation, that there's obviously so many other phenotypes that occur, at the motion segment, going just beyond the disc. We know that there's a string of different end plate abnormalities, defects, or whatever, modic changes, HIZs, et cetera, et cetera, et cetera. And we've done other studies looking at T1 Rho and how it relates to these different phenotypes. Have you looked at subgroup analysis of your subjects looking at these specific phenotypes? And do you think moving ahead as a society in the future, we should always just keep that in mind to consider these additional phenotypes when we think about the motion segment kind of going beyond just the disc itself?
Yeah, absolutely. I mean, we were specifically interested in biochemical changes, which is why we focused on the disc specifically and why we used T1 Rho in particular. But I think your point's well taken, that there are a number of non-disc related changes that are occurring, for example, at the end plates and the vertebra, the facets, the muscles, et cetera. And clearly those may also contribute to pain. So again, I just want to emphasize, we're not implying here that these patients had back pain that was discogenic in origin because we assessed disc degeneration. We're just saying that these patients had greater degree of disc degeneration than controls. So it may be that disc degeneration is like it's like the sine qua non of all the spinal degenerative disorders. It's not sufficient, but it is necessary, and it underlies a number of conditions that do cause pain, even if it doesn't cause pain itself.
I think we have to stop. I'm sorry, we're running out of time. So a big applause. And please, your co-workers that are here. Is there any co-workers? You stay, please stay, please stay. So can I ask all groups to come up? We want to have a photo. And while you are doing that, all three groups, prize winners, all prize winners here present in the room, please come up to the stage. And I want to thank you all for listening. And also I want to encourage everybody for next year to send in your best papers. And please use the video if you want to encourage people outside this room also to submit their work. We want to have the best papers to this prize. And I appreciate all the discussion here. So thank you, everybody.
And then we're going to have a 5-minute focus group report out. So if you want to hang here till these pictures are finished. That was good. Okay, so I'd like to ask the leaders of the focus groups to come up. So if you all already know, we have dedicated time in the program at the beginning of the week to have groups who are interested in specific important topics to come together, and these typically bridge from year to year, so there's progress made over time. So there was 3 of them that were held on Monday and Tuesday, and they're going to give a couple of words each just to let you know what the focus group is mostly interested in and their goals for the coming year. So maybe, we'll start in no particular order. We'll start with Jeremy, who has a focus group around automated spinal phenotypes.
Well, thank you very much for the opportunity of setting up this group, which really arose from the papers that you've just been hearing. So when I saw what everybody had done, I thought this is a good opportunity to pursue this. So what I'm really aiming at is to looking at the people who have collected large cohorts and to try and, to some extent, standardize how we describe these so that there's potential for pooling and sharing of data. So for the asymptomatic cohorts, it's really important that we're able to define what asymptomatic means, and I think that's been mentioned by the speakers already this morning. And on the symptomatic side is really to try and nail down and replicate, again, our findings, but the findings of the other groups and future studies as well. So that's progressing. We've got a website that's being born to deal with this, and I hope we can come back next year. So we'd like to make a booking for next year in Milan.
Okay. So the next one is spinal phenotypes. Gwen and Paul are going to tell you what we talked about.
So the phenotype precision spine focus group was a combination of the continuation of the phenotype group that's been going for a number of years and a new component related to precision care of spinal pain. The focus this year really was around trying to identify potential predictive biomarkers that can guide the treatment selection for patients with low back pain. The focus group had a number of parts. We're doing an expert consensus project looking for input from spine research experts and spine clinical experts to identify the factors that they believe may be responsible for or may predict the response to individual treatments. And so that project is a multi-phase project. We're currently in the phase of gathering information around the plausible treatments and the potential biomarkers or effect moderators associated with each of those treatments. The outcomes from the phenotype focus group, there will be an opinion paper that is written over the next few months trying to give a broad perspective of the problem. There'll be the outcome of this particular project related to the expert consensus of predictive biomarkers, and then there'll be another project related to trying to standardise some of the measures, and that relates to the second part of the focus group, and that was to have a report from the backpack group who I'll get Gwen Sauer to talk about now.
Thanks. I think what really came out thematically from the focus group was there's clear enthusiasm for some of this work, and so I really think we have a major call to action for this society. The backpack group, if you're not familiar with it, is a very large NIH-supported consortium in the United States. Well over $100 million has been invested in the last couple of years trying to get groups working together because, of course, we all have our own individual biomarkers that we're most interested in, but what's becoming increasingly clear is that we need really a conglomerate of those biomarkers to understand all the different domains of what's causing back pain and contributing to back pain and preventing response to treatment for back pain. So I think that what we've learned is that the harmonization part is one of the most challenging pieces, and I think that's where this society can really have a major impact in working together to create data sets that can be combined in meaningful ways, because if we're speaking the same language, it allows us to play that numbers game and really have a greater impact. I think the expert opinion piece is also going to be important, so Paul's work is incredibly important in this, and I happen to have a bias that I think this expert group with this society is really the key players that are going to make a difference in this, and the collective expertise that we have in various fields is just phenomenal. I think most importantly, I would say that what we've learned in this process and what I think we need to do to take the field forward is step out of our own comfort zones a little bit and not be trying to prove that our own individual biomarkers are the most important, but actually take unbiased approaches in working together. And that's where I think the collaboration is going to be most important to ensure that we learn what we don't know about back pain or even couldn't have even imagined. That might be an important trigger that's going to help us move the field forward. So I think it's really just an important call to action. So I hope many of you will get involved in this group as it moves forward. Thanks.
Thank you, Gwen and Paul. And the third was a long-standing focus group on degenerative spinal stenosis.
Thank you, Jeff. And I'm talking on behalf of Marcus Melloff, as well as Christine Tompkins-Link. Our focus group mainly this year is focused on the questionnaire that can be used by clinicians, how to use it to assess the treatment outcomes over time. So first of all, we actually find based on the systematic review published last year on what's the outcome measures for patients with lumbar spinal stenosis, and then we identified 20 questionnaires from that, and then after that we sent a survey to all the LSS members related to the focus group, and then we consolidated the findings and asking them whether they like the questionnaire or whether they would recommend the questionnaire or have they ever heard about the questionnaire, and then from that we finalized a list of questionnaire, and then during our focus group meeting we discussed them and then also asked for some other additional questionnaire can be included in future Delphi study. And for example, like patient expectations. And upcoming, we are going to ask ISSLS members regarding, we finalize the list and send it out, and then asking you, pick your brain, asking whether you think those questionnaires to be useful for clinicians in the future to assess patients with low back lumbar spinal stenosis, and also rank them so that we can know what should be recommended for clinicians before some new questionnaire can be developed. So this is, hopefully we will send out those survey very soon. Thank you very much.
Okay, so we're done for now. We'll come back at 10:30 for our next session on the fellowship report out. Thank you.